Hi everyone, I'm Sharanya. In this presentation, I'm going to talk about quantitative PCR. Quantitative PCR is an amplification-based technique for amplifying the DNA molecules in real time. This technique was developed by Higuchi in 1993. This procedure uses a modified thermal signal with UV detector and a charge coupled device that is CCD camera. The amplification and detection take place in a single tube, and this procedure eliminates post-PCR manipulations. There are several stages to this Qpcr reaction. The first one is exponential, second one is linear, the third one is Plateau stage. In the exponential stage, the PCR reagents are abundant and amplicons are doubled. In the linear stage, reagents run off and the reaction slows down. In the plateau stage, there is no increase in the amount of the product. The plateau stage is limited by the amount of primers, reagents, stack polymerase activity, and also re-annealing of product strand. The threshold line is a level of detection in a reaction in which the fluorescent intensity is above the background level. PCR cycle at which the sample reaches this threshold level is called cycle threshold or CT value. There are several detection methods for Qpcr reaction. The first one is non-specific cybergreen detection method. The cybergreen binds to minor groove of double-stranded DNA. Fluorescence increases with the concentration of double-stranded DNA. For cybergreen detection method, melting point curve analysis is formed followed by real-time PCR. The inflection point is a curve which indicates the melting point of amplicon. Contaminating DNA and primer dimers are seen as additional peaks. The third detection method of qPCR is by using molecular beacons. Fluorescence increases as the quench probe hybridizes to the DNA template. Molecular beacons are the double-stranded hairpin loop-like structure sequence that keep reporter and the quencher dyes very close to each other. During the denaturation, the beacons anneal to the target template, separating both dyes, and fluorescence increases with the concentration of the DNA. The fourth detection method is by using plexus system. Plexus system is a novel real-time PCR system used for quantifying the human and Y chromosomal DNA. One of the primer contains a modified nucleotide, that is iso-DC, attached with fluorescent dye at the 5' end, and the second primer is unlabeled. The reaction mix contains isodapsyl DGP and deoxynucleotides. The isodapsyl DGTP incorporates with iso-DC residue in the primer. The incorporation results in the quenching of fluorescent dye on the complementary strand. That is, the reduction occurs in the fluorescence and also amplification occurs with quantification in real time. The more the double-stranded DNA is produced, the amount of fluorescence decreases in the plexus system.